So today we're going to talk about the work that we've been doing over the last dozen or so years to develop a first principles view of molecular structure and function and mechanism that is kind of embodied in a theory that we call biodynamics. And it's not a rabbit out of a hat. This is something that we built from a lot of different contributions, and we'll give some credit to our contributors in a little while. But we're going to give you a whirlwind tour through this because this is very new science. We have to give a lot of background to understand what you'll be looking at in VR in a few minutes, so that you'll be able to make sense out of the whole story. And we're going to exemplify our work by way of COVID Mpro. So we published a paper in 2020. Hongbin and I did a deep dive on Mpro structure function, like trying to decipher the whole catalytic cycle of the enzyme using our theoretical principles, which we call biodynamics. And this covers the atomic level of resolution as well as the systems level of resolution that you'll see in this paper. So hopefully everyone will have a look at it. So we'll start out kind of with a big picture view of biodynamics, which again is a theory, it's first principles, it's not based on empirical science or data modeling in pretty much any way, shape, or form. So it is something that is very novel. But it's a framework for understanding how molecules and molecular systems work and how molecular systems deliver cellular functions. So you can see we have this zoomed in atomic scale resolution level, and then we have a zoomed out systems level resolution scale. So biodynamics is a theoretical framework for understanding the actual inner workings of molecular and cell biology and molecular and cellular dysfunction. And so we're dealing with the zoomed in level of understanding at the atomic scale, and we're dealing with the zoomed out level at the multimolecular scale. So the atomic scale, it's more about molecular structure and function and how this is powered. And at the systems level, it's really how all of this plays together at the molecular level into molecular systems that define how cells actually think, so to speak. And we're working at the intersection between physics, biology, and chemistry. So that's kind of, I think, what sets biodynamics apart, is in drawing from all of these different areas and integrating them into sort of one scientific platform. So where we're going to focus on today is the atomic scale, and particularly how molecular structure and function are powered. And classically, people think about free energy in terms of, I'm talking about non-covalent free energy, in terms of contacts, so interatomic contacts like hydrogen bonds, van der Waals, hydrophobic, etc., that we're all familiar with. It's all in the textbooks. It's all what we're taught in school. But it seems to be that this thinking is actually wrong. There is energy in these contributions. There's no question about that. But that's not the predominant energy. The predominant energy is actually in the solvating water. And this was an idea that was advanced by Dan McKay and Tom Kurtzman several years ago. They were the first to make this claim, to the best of my knowledge. And it's kind of a philosophy about how to think about solvation, the mechanisms of how solvation free energy is used to drive molecular structure and function. So it's kind of a philosophy, which is not to say that other groups have not been working in this area, but again, it's a philosophical difference in this philosophy that we really agree with and have been expanding considerably and applying it considerably, in particular connecting the dots between solvation free energy and kinetic free energy barriers, which we published in 2010. So why do we think that the lion's share of free energy is concentrated in water, in solvating water? And the reason is shown in this plot. This plot was something that we had published, I think, in 2015 or something around there. Work that was done by my colleagues Camillo and Dan. And what they did is they plotted the melting point and the boiling point for a large number of solvents. And so organic solvents, a wide range of different types of solvents, as sort of to get a perspective on where water sits relative to everything else, pretty much. And you can see that it's like the difference between the Earth and the Moon, distance-wise. Water is way out on its own in terms of its free energy density. So it's energy per unit volume. How much energy can you pack into a 30 cubic angstrom volume that can go into all the nooks and crannies around amino acids or in folded proteins and so forth? Water is clearly the only game in town for this. There's nothing that even comes close. And so that's how we get the scale that we have, a microscopic scale, how the sizes of biomolecules that can fit into a cellular volume, basically, is because of this high energy density that water brings into the mix. And in fact, water is the energy is so strong that it perturbing it can basically be at the level of a single carbon, and the water knows. It's not like in the noise. It's actually a strong signal that water knows that it's being perturbed just by one single carbon difference between different solutes. So it's both a solvent and it's also an energy source at the same time. So how does this work? So in order to actually harness the solvation energy, we have to be able to store energy into the solvation and release energy from the solvation. And the way this works is that when a solute is introduced to water, to pure water, that the free energy of that water actually is split across the surface of the solute. So from one point of the solute to another, to another, to another, we see that some of the water is hydrogen bond depleted. So it's losing hydrogen bonds relative to bulk solvent, which is 3.5 hydrogen bonds per water. They're moderately strong, so we can actually lose hydrogen bonds or hydrogen bond strength. Why? Because a particular position on a solute surface doesn't have polar groups. So there's no hydrogen bond partners, or there's fewer hydrogen bond partners than water would find in bulk solvent. So that water becomes depleted at that particular position. And then we have the other end of the spectrum, is that that water can find more hydrogen bond partners in the solvation than in bulk solvent. And so that water then becomes hydrogen bond enriched. And there's a whole spectrum in between, including bulk-like solvation, which doesn't really matter in terms of energy. It's on these opposite ends of the spectrum that really count in terms of driving or powering structure function, as we say. So again, this is first principles. The ideas that we're talking about here are not about measuring anything and fitting data to build models. This is all with the underlying assumption that the lion's share of non-covalent free energy is stored in solvating water. And so then the question is how do we think about this? Well, we started to think about it in terms of a field. So hydrogen-bonded water that is solvating a given solute on the surface you can think about this in terms of the network of those hydrogen bonds that are wrapping around the solute. There are going to be bumps and valleys or hills and valleys in the terrain of that surface, and the water is trying to conform to those hills and valleys, and so the hydrogen bonds may be distorted in some places and really well defined in other places. So it's sort of a field of hydrogen bonds that can be perturbed by the solute that it's solvating, as well as by other solutes that come along to bind. So what you're seeing in this slide is essentially a sampling of this hydrogen bond field on the surface of Mpro in this particular case. And we're using the water occupancy at each of these grid points, or these lattice points, as a surrogate for the hydrogen bond quality at each of these points. So where the hydrogen bond quality is high relative to bulk solvent, you'll see a larger sphere at that point. And where the hydrogen bond quality is lower, you'll see a smaller sphere. So it's a whole spectrum of sphere sizes, which is not something you have to worry about in this particular slide. It's just the point is that we're sampling along a grid. We're sampling that hydrogen bond field to find where there's depletion and where there's enrichment.
Bob, I have a question.
Yes,
how does
sure.
this, or does it even, relate to the solvent-accessible surface area that people like to compute?
Well, again, this is a question of where the water can go. So if you play with the radius of the water to smooth out the surface, then you create the solvent-accessible surface, right? Or you could have a van der Waals surface where you don't smooth it out. You just think about the water as radius as small enough that it can go into all the nooks and crannies. So really, at the end of the day, it's about where water actually can go into some confined space, and how many hydrogen bonds it's getting in that particular space. If you were to look at the actual donors and acceptors in the environment of that water, so we're not really playing a game about solvent-accessible surface. We're just saying let the water go where it wants to go. And we do this during an MD simulation where we're focused on the water, not on the structural rearrangements, but how the water is moving between bulk solvent and the surface of the solute of interest, and the water just goes where it wants to go. So we don't have to play any games with the surface representation. Does that?
So it has more to do with like the potential energy of the water?
Right. Well, there will be, there's a steric repulsion. The water can't go into the protein surface, right, or can't go through atoms. But in terms of where it wants to go, where you'll see higher occupancy water in the simulation, is based on the quality, strength, number of hydrogen bonds that it makes with the local environment on a particular position of the protein surface. That make sense?
Thanks.
Okay. So that's kind of the gist of what we're looking at in terms of the solvation fields. And we're going so far as to say that the solvation fields, which again the hydrogen bond network and the way that it responds to perturbations, that this is the puppet master of solute structure and function. So it's not that the structure decides how to fold, like a protein would decide how it's going to fold or what it's going to interact with. It's its solvation is basically directing that. The water is trying to always maximize its hydrogen bond number and strength, so minimize its hydrogen bond free energy. as much as can, because it's such a hydrogen bonding pig, really, that it won't allow solutes to steal its hydrogen bonds away. And if a solute steals away too many hydrogen bonds or weakens them too much, that solute will crash out of solution. Water always has the last word in what goes on. Okay, so the point is that we can store positive free energy in certain regions of the protein surface, and we can have negative free energy. So in other words, the energy is higher than in bulk solvent is positive, is lower than bulk solvent is negative, and we can have different regions of a protein surface that have that range from positive to negative, which is exactly what we see. And where there's positive free energy is like rolling a ball up a hill. You've stored energy in there. Now you can release that energy to do work. So that's the really important point here, is you have to have positive free energy in order to do work. That's what's getting released. And it's very difficult to explain how you get positive free energy stored in interatomic contacts. I mean, it's very straightforward to understand this with solvation and not with interatomic contacts. So now we're talking about everything from protein folding all the way to membrane formation and crystallization, permeation. Everything that happens in an aqueous environment that is non-covalent is being powered principally by solvation free energy, and in particular by desolvation costs and resolvation costs. And we're going to get into that in a minute. So we talk about how H-bond depleted and enriched solvation are distributed over a solute surface, but it turns out that that's not random. There's specific patterns of these distributions, which are shown in this cartoon. As the free energy of water, it is a function of position on the surface, that the water is going, the free energy of the water is going positive in some regions, negative in others, or around the bulk solvent level in others, and it's going very negative, say here, very positive, say here, okay, and that this is all very non-isotropic and very non-random. So again, it's the solvation field and how the solvation field, how energy is stored in that field from position to position going around the surface of a solute. And so you could see again here, there's certain places where the water really would be low occupancy, depleted of hydrogen bonds with the lightning bolts, and then others where the water is actually enriched in hydrogen bonds. So it shows up in the occupancy of that water at each of those positions. The occupancy is low where it's losing hydrogen bonds, and it's high where it's gaining hydrogen bonds. Now we call this pattern the solvophore, or the inverse solvophore. In the case of if you want to define a binding site, would have an inverse solvophore that would recognize the solvophore of some cognate partners, whether they're other proteins or low molecular weight. So that's the idea here, the solvophore.
How does that representation differ from, some of the other tools people are using these days, like, water map or water flap, just in concept?
Yeah, that's a great question. So again, we're not focusing on the tool here. We do have the tool, which we'll be talking about in a second, but we're focusing on the science. So the concepts, what's behind the tools, and whether the tools really capture the science well enough to actually guide us in doing drug discovery and understanding molecular structure and function. So we can, argue about whose tool is better, whose tool captures the reality better. But one thing I can say is that the tool that we're using uses unrestrained MD simulation. So the protein is free to rearrange within the 100 nanosecond time scale, and the water is free, obviously, to rearrange. And other approaches, tend to restrain the protein, which we find actually is a serious limitation. The water does not behave in its native way when the protein is restrained. It's very sensitive to that sort of thing. So yeah, it's both the technology, but it's also the thinking behind it where we differ.
Right. Great.
So this is the idea of inverse solvophore. So inverse solvophore would, say, be a binding site, and a solvophore would be, say, a ligand, a small molecule could be. And in this case, you see that there's poor matching between polar groups on one side and nonpolar groups on the other side. So basically, you end up with either sort of clashing between different mismatches, say, between the solvophore and the inverse solvophore. So we want polar groups matched to hydrogen bond enriched solvating water on the opposite partner, and we want nonpolar groups matched to hydrogen bond depleted solvation on the other partner in both directions. So the hydrogen bond enriched water, solvating water, we call those gatekeepers, because in order for binding partners to approach each other and have surface contact, they have to go through the hydrogen bond enriched water, which means they have to replace the hydrogen bonds of that water to expel that water at low cost. That's why we talk about desolvation cost. The higher the desolvation cost means that the gatekeeper is very strong in preventing, say, molecules from binding, or the gatekeeper is weakened by replacing the water hydrogen bonds of the expelled water. And then for the molecules to dissociate, which would be the dissociation barrier, is really largely the resolvation cost of the hydrogen bond depleted positions. So in other words, bringing water back to those unfavorable positions, that cost is the dissociation barrier. So it ends up being sort of the snare. So if molecules desolvate hydrogen bond depleted solvation, it keeps them bound longer. So the lifetime of the bound state is increased that way. And so we can talk about the relation to K on and K off simply in terms of desolvation and resolvation costs. And we've been testing this idea for years now, and with real measured kinetics data, and it works very, very well. So if we're saying that the free energy, non-covalent free energy, is stored largely in solvation, then we have to start thinking about another level in the omics hierarchy that we're calling the solvome. So this is, its form follows function, follows free energy. And this is where the free energy is stored here. So it's not just the structures going from gene to the genome to the proteome, etc. It's also how these things shape the free energy that's available to power rearrangements, functional rearrangements, as well as intermolecular interactions, so binding interactions, say between proteins and ligands or enzymes and substrates and so forth. So in our deep dive on Mpro in 2020, we actually really zoomed out and we were looking at pretty much the entire catalytic cycle. I don't have time to go into that here, but I just wanted to mention that biodynamics has this zoomed out, zoomed in capability to look at specific rearrangements as well as a global set of rearrangements that define the overall structure function of the protein. And we're doing this with other proteins as well. So our technology for mapping the solvation field is called watMD. So it's software that was written by Dan McKay and Vivas Aravimathan when they were at Novartis. And so again, this is predicting hydrogen bond enriched and depleted positions around a lattice, which you can see is sort of idealized here. The protein sitting in essentially a box, which is filled with water. The box is artificially partitioned into cubes, which we call voxels. There are only 1 cubic angstrom in each dimension, which avoids having more than 1 water molecule per voxel, or even more than 1 atom of a water molecule per voxel. Because at the end, we're counting atomic solvation. So hydrogen versus oxygen occupancy in these voxels or cubes is what we're counting. And you can see that the counts follow a Gaussian distribution. If you look at the overall surface around a given solute, it's a Gaussian distribution of occupancies, which again mirrors the free energy of that water and the hydrogen bond enrichment and depletion. So the depleted water is way out here in the left tail. That's what we really care about. And the enriched water is way over here in the right tail, is what we care about on that side. Nothing in the middle, because there's not much energy that's being stored, either positive or negative energy in the middle. This is more in the bulk-like solvent regime. So we have the Gaussian distribution for hydrogens and the Gaussian distribution for oxygens visiting the cells, and we're really looking at the cells that are right up against the surface of the solute. Okay. Oops. Okay. So now we want to focus in on a particular example where we have the inverse solvophore is in the catalytic site of Mpro, and then the solvophore of an inhibitor, which now we're saying that what you're looking at is not just a collection of spheres that were computed, but is actually the horse's mouth of SAR, or structure activity relationships. This is where the energy is. This is how the molecules see each other. And I'm going to turn this over to Hongbin now to show you in VR exactly what we have relative to Pfizer's, which is now kind of a famous Mpro inhibitor drug, and show you why that compound actually is as good enough to become a drug in the first place. And I'll turn it over to him now.
So our paper, we studied several Mpro inhibitors, include the one that Pfizer published last year as a drug analog. And then our paper, we would like to see the complementarity between the ligands and the inverse solvophore from our calculations. Today we will focus on the match in the catalytic site, including how the polar groups are positioning toward a hydrogen bond enriched solvation positions, and non-polar groups at hydrogen bond depleted positions, and meanwhile also how the ligand trying to avoid the non-polar hydrogen bond enriched and polar hydrogen bond depleted mismatches. So let's grab the structure here. And here's the solvation field we calculated for Mpro inhibitor. And you can see here a lot of voxels spreading everywhere. And the smaller sizes are representing the low occupancy voxels, and then larger ones are representing high occupancy voxels. And then color red or reddish color representing the oxygen atom favorable positions, and then the blue light color are representing hydrogen atom favorable positions. So the ligand here are the Pfizer's drug like analog. So let me hide the voxel here. You can see in the P1 pocket, they have a cyclic amide group nitrogen here from the glutamine.
Right.
And then, so our predicted voxel here, you can see the amide nitrogen overlays really well on the high occupancy voxels. And then see here, in the S2 pocket, so P2 pocket, you see here the carbonyl group overlaps with the high occupancy voxels really well. And plus, in the end of the molecule, you see here has an amide group here, also overlay the high occupancy voxels really well. And then here are all those favorable ones, but you can see the rest of the two rings structure, they do not overlay the high occupancy at all. Instead, they wrapping around with so tiny voxels here, so which are contributing to the slow K-off, because those are the low occupancy voxels predicted. Let's open up that drug. So from Pfizer, so we overlay the real drug candidate from Pfizer and color them in blue. So comparing to the analog, the majority of those structures overlay really well, except here, the S3 or S4 pocket. You will see here has the three fluorine groups here. Right. They clash with the high occupancy voxels, which is unfavorable. So that's kind of well explained why the drug candidate has around 10-fold less potency comparing to the drug analog.
Sure. Yep.
And I guess in the very end, the Pfizer preferred to go through the drug in the end because of the bioavailability. Yeah. So speaking of all the spreading out of the spreaded low occupancy voxels, technically we can grow the ligands more and more to make it like to gain more hydrogen solvation free energy, but that may lose a lot of the admin properties also. So that's so we have to think about how to balance the free energy gain and also the ligand properties.
Having a more direct assessment of the free energy contributions for binding, it gives you a lot more control over your chemistry, because now you're not just doing this trial and error kind of
approach,
compound design, but you're really looking at the free energy, the real free energy sources, and so now as long as you keep everything in register with the inverse solvophore, that you're free to play with other parts of the molecule that will not disrupt that. Whereas with traditional methods, it's sort of a free-for-all, because you're not getting the right feedback about the structure-free energy relationship. So that's a guessing game, and then everything else is a guessing game. So it ends up being nothing more than trial and error at that point.
Yeah, makes a lot of sense. Yeah, excellent work and certainly a revolutionary way of thinking about the problem. You can see the power of it, definitely. And helping to visualize it here at Nanome, is certainly also very powerful.
Well, that's a good point, Carla, because we're not calculating numbers. We don't think that you can correctly calculate numbers, not by any kind of force field-based methods, free energy perturbation methods, you name it. It's just because the free energy is in the solvation, we have to kind of limit ourselves really to that contribution, or focus on that contribution, I should say, the most. And so we're talking about visualization. The whole thing is based on visualization. So where are the voxels? What size are the voxels? Where are they? How would the water be oriented in each of these clusters of voxels? These are not water molecules. These are positions where water is hydrogen bond enriched. But you could see that where there's blue, that means there's a preference for hydrogen. Where there's red, there's preference for oxygen. And so you can imagine a water molecule sitting in this cluster that is maybe going into this position from bulk solvent in a particular orientation one time, right, and colliding the next time in a different orientation, or is just sort of wiggling around in there, trying to maximize the hydrogen bonds. So it's all visualization, and VR is the only way to go. If people are trying to look at this using just mono visualization, rotating the molecule on the screen or looking at a static image, there's just no way to get the three-dimensional depth perception that you really need to use this to design compounds.
It definitely speaks to the power of Nanome for drug discovery projects.
Exactly.
For sure.
It's getting the science right and getting the visualization to be everything that it can be. And so you're getting the chocolate and the peanut butter, really, here.